it's Mrs. Shannon, and we're going to go through the cell division Prezi. Please watch this one before you go into the cell cycle regulation. This is going to give you the background of what the cell cycle actually is and how cells divide. The cell cycle regulation goes in more to what signals prompt the movement through the cell cycle and what happens when these signals are not functioning correctly. So let me go ahead and put this in big screen. And we'll go ahead and start. This presentation specifically are going to address 2 points. Number 1, why do cells need to divide in the first place? What's the whole point of cell division? And number 2, how does cell division provide for a continuity of life processes, both within the individual itself, which means it's going to allow these processes to go on within an individual, but as well as in a species. And so when we're looking at why cells divide, we're going to first look at the continuity of life. So in order for an organism to survive, individuals must replace any damaged cells. So if it's damage to physical issues, scrapes, burns, if it's just age itself, cells need to be replaced. If cells are not replaced, if damaged cells are not replaced, then the organism may not survive. In order to grow, the number of cells produced must be greater than the number of cells that die. So, in order to survive, the species must replace individuals. Individual cells need to be replaced within individuals, but for the species itself, individuals, especially in single-celled organisms, need to be replaced through cell division. So cell division accomplishes this, and the way that a cell divides is a function of the cell cycle. There are different phases of a cell's life, just like a person has a life, or a plant has a life cycle, or another animal has a life cycle. There are different stages or phases. In G1, we're looking at our growth phase. This is where normal growth happens. It's getting larger, normal metabolism. In the S phase, we have DNA replication, because the idea here is that if we are going to divide from one cell into 2 cells, we are going to need to be able to have exact replicas, exact copies of the DNA that are going to be able to be placed in both cells. After that happens, we enter a G2 phase. This is like another growth cycle, but it's more growing, making sure we're healthy, doing any kind of last minute catches or repairs before we go on into our M stage, which is mitosis. Mitosis is the cell division itself. So you can see here, just looking at this, it's a relatively short cycle. If a cell is not going to divide, what happens is during this first growth phase, it will actually leave the cell cycle and it will go into what we call G0. G0 is a non-dividing state. I call it the waiting room or the lounge. Most cells in you are not dividing or going to divide, and so they are in this little lounge here. And I call it a lounge because there are times that some cells will enter back into the cell cycle, like the liver cell. And so here you can see we have one cell going on through the cell cycle. It's going to divide, produce 2 cells, and each cell, actually we could have another cell cycle going on up here. Each cell is going to then follow through the next cell cycle again. Now, in order to divide, in order to continue on with this cycle, we're going to have to replicate our DNA. And in order to do that, we're going to first have to form chromosomes. Chromosomes are tightly coiled pieces of DNA, and they condense prior to division. The idea is that they condense and they tighten up, because otherwise what would happen is you'd have this entangled mass of DNA trying to replicate and divide, and then the chance of damage to them will be much higher. Most eukaryotic cells have 2 copies of every chromosome. And so here's a tightly coiled chromosome right here, a tightly coiled DNA, and we have our 2 copies, and they are attached with a centromere in the middle. Chromatid is what we call one member of the pair, and the centromere is the region that they attach to. And whoopsie, let me go back up here. Don't know how to get that little thing off. If that little screen at the bottom is bothering you, I'm sorry about that. Just as a side note, remember, prokaryotes only have one circular chromosomes. They don't have the same issues that we do in other eukaryotic organisms. Eukaryotes have many linear chromosomes. So there are going to be some changes when it comes to cell division because of that. For instance, in this prokaryote, you have one circular strand of DNA. During DNA replication, we only have two circular strands. It's a lot easier to segregate. Okay, it's a lot easier to segregate, and this process right here is known as binary fission. But with all of our linear chromosomes, there's going to be a little bit more of an extra step involved. Make sure that you do understand the chromosome-chromatid relationship. It can be confusing. We're going to spend some time second semester looking at this whole relationship with DNA, and we're going to model it from DNA to chromatin to chromatid, chromosome, and identify the different parts of it, okay? So that's just something to keep in mind. So here we have our double helix right here, our DNA, chromosomal DNA molecules. They replicate in the S phase, and during mitosis, they divide back. And so you start with one here. You replicate into two copies, and then when you divide, each cell will get one. So we're back to identical copies of what we started with. Haploid versus diploid, in case you don't remember these terms. Haploid means one copy of every chromosome, and we use the lowercase n to represent that number. Diploid means two copies, and we use 2n to represent that. Human cells have 23 pairs of chromosomes, which means that we are diploid. 23 times 2, 46. Okay? So how many chromatids are present during G1 and how many chromatids are present during G2? So G1, we have our 23 pairs, 46 chromosomes. G2, we should realize that we have 96 pairs, 46 pairs or 92 chromatids. Now, mitosis is non-reductive eukaryotic cell division. That means whatever we start with, we end up with the same amount of DNA. And that's going to be different than meiosis, which we'll look at later. So during interphase, we have a growth of the cell still going on, especially in that G1. We have replication of DNA in that S phase, and we have preparation for division during G2. Most of the cell's life cycle is in this phase right here. And so you can see that they are in this normal stage, or this interphase stage, because you can see I call it a blob, a little blob where the nucleus is, but you can't really make out the chromosomes too much. You can see a definite nuclear envelope. It's still in the sort of central location of the cell. And so this is interphase. During prophase, the chromosomes condense. Now remember, they were already condensed to begin with. They were already circular spiral DNA that was condensed to help avoid errors in replication, but now they're going to condense even more so. And when they condense, you can start to see them a little bit more under the microscope because they become more dense underneath the microscope. The nuclear envelope breaks down and mitotic spindles begin to form. Now, prometaphase, the difference between the two is that the nuclear envelope is going to completely disappear at this point. And the chromosomes are actually going to start moving. You should be able to see definite strands of chromosomes at this point. When they're in metaphase, I always think of they meet in the middle. I met up with my friends in the middle of the cell. I met with my friends. They are at what's called the metaphase plate, and the spindles attached to the kinetochore of chromosomes. And so the spindles are these little green lines here. You can see the green lines. You can see the blue chromosomes. And the spindles, it's like a little spider's web. They attach to the center of these chromosomes. And here you can see beautiful examples of metaphase. You can see how they're lined up here at the plate. Okay, and you can actually see the connection of the spindles, the kinetochore microtubules, the spindles hitting the kinetochore. In anaphase, they start being pulled away, the chromosomes, or the chromatids. The chromatids of the chromosomes start to split apart at the centromere. Okay, so here's sort of a diagram here. We have our spindle pull, we have our kinetochore, and we have the spindles pulling them away. And in telophase, we have the chromosomes start to decondense. That means that they start to loosen back up, and then the nuclear envelope starts to reform. Cytokinesis is the final step, and that is when the cell membrane actually divides. So once this is completely separated and it pinches off here, we have a cell membrane that completely pinches off, or in a cell wall that reforms if we're talking about plant cells. That's when we have cytokinesis. So here you can see that the cell plate is starting to form. It's not, you can still see some of these tubules though, some of these spindle fibers still here, so it's not complete. Cytokinesis is not complete, but I would say that telophase is easy to see at this point. So there are a couple of differences between plant-like and animal-like cells, the reason being that host cell wall. Cytokinesis is the splitting of the cell into two. And so with animals, we sort of have a contractile ring of microfilaments that sort of pinches it off. And you can see it right here. It kind of just pinches it off. In plants, however, we have that cell wall. And so vesicles from both cells, both of these new cells developing, deposit part of a cell wall here that's going to fuse together. Now, when it comes to the rest of the organelles, we're focusing a lot on chromosomes and DNA and the nucleus, but when it comes to the rest of the organelles separating themselves, it's essentially a random process. There is a little bit of research that has found that maybe there is organization, especially when you're looking at like the mitochondria and how many and where they end up, that there is some control, but not much is known actually about how organelles disperse themselves. So here's a mitosis at a glance, and let's play spot the phases. And so these are like cards that you'll be using. And so if I were to point to this phase right here, hopefully you guys can see that it is anaphase. If we were to do this one right here, hopefully you guys can see that it's prophase because we can see it condensing up. This one right here is metaphase. We're meeting up in the middle. This one, actually, I would probably call this one a prometaphase, and I would call this one right here a prophase itself. These over here, you should be able to identify as interphase. Let's see here. We have a telophase going on right here, and it looks like cytokinesis hasn't really started at all. This is just the onset of telophase. Whereas up here, you should be able to see that the cell wall is very faint, but it's already forming. Cytokinesis is going on. So you should be able to look at different images and spot which phase it is when it comes to cell division, or if it's just an interphase here. So watch it happen. I don't know if that's going to be working. It's not working right now, so I'm not going to be able to get it from here. So here's mitosis in real time. And so you can actually see the cell division actually going on. You can see the spindle fibers. You can see the chromosomes. Okay. So I'll play that one more time here so you can see the actual condensing of the chroma. Oh, we're condensed. Here we go. Right. There we go. We had our centrioles forming. Whoopsie. So the centrioles are up here. We have our spindle fibers forming right there. And I keep stopping this, and I should just let it go on. But if you're in the actual Prezi itself, you should be able to play this without having it stop in different locations and keep repeating it. And you can see the two nuclei and then the division of the cytoplasm at the end. So, of course, we tie everything back to why is this important? How does this connect to other organisms? Is there a connection between other organisms? And when we say other organisms, we're talking about other species. What are some similarities and differences? And so there are more similarities between eukaryotic and prokaryotic cell division than might be apparent at first glance. And so when we talked about like binary fission versus mitosis, we talked about circular DNA, one copy versus lots of linear copies. At first, they seem pretty far-fetched, but far off. However, proteins involved in binary fission and eukaryotic cell division have a large degree of homology. Homology is a term that we'll see a lot when we start talking more about evolution. Homology means similarities, same origins. And so when we're talking about these proteins, these are the proteins that signal cell division to occur, that signal the cell to enter different parts of the cell cycle. So these controlling and regulatory proteins are very similar, have very similar origins in both prokaryotic and eukaryotic cell division. And in fact, there are also some unicellular eukaryotes that demonstrate sort of this like mixed modes of division, where it seems like diatoms and yeast here, where it's kind of like a combination of binary fission and mitosis in one. And so these intermediates here, and the fact that we have regulatory proteins that have a large degree of similarities, homology in the genetic sequence, origin of the genetic sequence, indicates that there is some consistency, there is a relationship between the processes in different organisms. So I hope this helps out. Again, I hope you watch this before you do the regulation. It gives some vocabulary. It also sort of illustrates the different phases. At this point, you should be able to make sure that you can describe the roles that mitosis plays in eukaryotic organisms. So that ability to retain the continuity of life within the organism, to replace damaged cells, to replace dead cells. Explain how mitosis produces two genetically identical cells. Explain how interphase prepares a cell for mitosis. And so when we're talking about that, we're talking about the condensing, we're talking about the synthesis of the DNA itself. We're also going to be talking in the next Prezi about regulation of the cell cycle and checkpoints that it's going to go through. Explain why many cells never divide. And so those are cells like the liver cell, the nerve cell. They don't need to divide. Their energy is being stored into or being used for what their job actually is. And then compare the events of mitosis in plant-like and animal-like cells. And so a big part of that is the whole cytokinesis and the cell wall formation versus the pinching off of the cell membrane in the animal-like cells. Now, there's one term that we did not go through, and that is apoptosis. Sometimes, and don't think it came up in the cell regulation one, sometimes if a cell is unable to divide, if there is an error and it's not able to divide, a cell will self-destruct, and that is called apoptosis. It's controlled cell suicide. And so what happens at that point is that signals, if the cell is unable to continue through the cell cycle for any reason or any errors are caught that prevent it from entering into mitosis, the cell will undergo cell death, and that will prevent errors in new cells that are being formed. So I hope that this helps you. Let me know if you have any questions.